Welcome back to the lecture series in cell culture technology. So we are starting the fourth week. So this week, in the initial part, we will be covering about the different kind of extracellular matrix. So if you recollect in the last week, while we were discussing about the role of extracellular matrix, we talked about the kind of developmental aspects and the way the extracellular matrix anchor the cells on the substrate. So today what we will do, we will first of all enumerate the different kind of synthetic and natural extracellular matrix which are currently being used in the cell culture technology. And we will talk about the possible interactions what these extracellular matrix materials are conferring on the attaching cell. So, to start off with lecture 1, and we are into week 4. W4/L1. So, to start off with, we will be talking about different types of extracellular matrix used in cell culture. So let's classify them, as we are mentioning, into two broad categories. Okay. Synthetic and natural. So when we talk about natural, we talk about the one which are known to show this effect. Either these could be obtained from natural sources directly or using biotechnological approach. They could be produced in mass by transferring their genes into some other secretory protein cell lines where you can produce them. Okay. So talking about the natural ones to start off with, we are aware about collagen, matrix protein, extracellular matrix protein, which is very dominant in ensuring the anchorage between cell to cell and cell to other cell type, and collagen has several types. So we are not getting into the types, but under the broad umbrella of collagen. Then we have laminin. The cells have integrin receptors, and these laminins bind to the integrin. We'll come to that. Okay. Then in the same family we have a mix kind of a stuff of collagen, laminin called gelatin. Okay. Whereas in the natural or the synthetic side, we have poly lysine, we have this one. I will giving you some literature on this one. We will talk about this. This is basically a silane, DTA silane. These are tetramine groups which are present in them. We'll come later into that. So let's start our discussion with these different synthetic and natural extracellular matrix. So talking about the synthetic matrix, it is easy to talk about the synthetic matrix because most of them do not have any specific interaction such. So the way say polydilysine works is something like this. So, for example, here you have a substrate, a glass substrate. On a glass substrate, you deposit polydilysine. Deposit polydilysine. So, talking about polydilysine, D is the isomer type and lysine, as you know, is an amino acid. So, we have a chain of lysine. So, if this is the lysine residue, attached with peptide bonds out here, out here, out here. And if you buy this polydilysine from the market, so it comes as polydilysine hydrobromide, that is to stabilize it. It will come like HBr. But do not worry, that thing doesn't remain in the culture, it kind of removes away. But this is essentially the actual molecule is. And while you put it on the surface, the molecules become something like this. Forms a fairly uneven surface, because if you take an AFM image, I will share an AFM image of this as an additional information. So, it's something become like this, and if you take the AFM tip to do a surface analysis, then what you will see is something like this. It's a very, very kind of rough surface upon surface analysis, and they are exposed. So, this has bunch of NH3 groups, positive NH3 groups, which are popping out. These NH3 groups are like this, they are popping out like this from the amine terminal. Okay. So, if you remember, so this is how your amino acids will look like. It has COO terminal on one side, it is amino group on the other side, and it has a R group here, and it has a H here, right. So, it has few of these amino groups which are present here also, few of these amino groups present here also, okay. And these ones kind of pops out. So, what you see essentially, the surface, by some kind of a surface interaction on the glass surface, they attach. But before you can put them on the substrate, what you have to do is, you have to really clean the glass substrate very thoroughly. Whenever you do this kind, because these kind of, it is precisely a process of adsorption. Depositing essentially means you are adsorbing on the surface. You are not doing any chemical coupling of the polydilysine molecule on the substrate. So, what you do is, you take polydilysine in a vial, you mix deionized water into it, and you dissolve it thoroughly, and in that process you get a solution. So, the concentration is generally used for polydilysine is fairly low concentration, okay. And the exact concentration I will provide through few published literature. It has to be on a lower side. So, always remember, one catch about polydilysine is that polydilysine at a higher concentration does not promote cell growth. Poly-D-lysine at a fairly low concentration does promote cell growth, but good enough to make almost like the surface coverage should be full. So you have to keep that in mind. The surface coverage should be completely full. Leaving aside, of course, there may be little space out there that you cannot really judge unless otherwise you do atomic force microscopy on it. And the second thing what you can do to analyze the surface, so this is how we'll be proceeding. Okay. First, you should do AFM, atomic force microscopy, to analyze the surface. When you have to really learn, and once you exactly learn with that particular composition, the surface will look like this, then you can repeat it, but time to time you have to cross check. Okay. First, you should do a physical surface analysis. Second thing, what is essential is put a drop of water on the surface and see how the drop of water upon falling on the substrate kind of spread out. Does it remain like this, or does it spread out like this, or does it spread out like this? So based on that, you measure the scientific term what we call as the contact angle. It may be like this, it may be like this, or it may be very broad like this. Okay. So this is called contact angle measurements. This contact angle measurements will give you a fairly good idea about the hydrophobic and hydrophilic nature of the substrate. Hydrophobic or hydrophilic. How? Even without getting into the details, you can see if, so all of you have seen this picture, you must have all seen, right? Suppose there is a leaf and there is a water droplet on it. The water droplet are like this. So whenever we see a water droplet like this, what is our common sense? It says that the water molecule is not really flattening out. It means this substrate or this surface of the leaf is hydrophobic, right? It does not allow the water molecules, the contact point between the water molecule and the leaf is very, very minuscule or minimum. Okay. So you can really measure the angle just by looking at this picture. You can see the angle will be something like this. Very little contact out there, narrow contact. On the contrary, say for example, there are substrate where you put the water molecule, suppose this is the substrate, and you put the water molecule almost in no times become like this from here to here. We call them fairly hydrophilic substrate. The water can really spread out very fast. Now based on this, I can suggest you an experiment if you try. If you increase the concentration of poly-D-lysine on the substrate, say for example, here I said about low concentration, say for example, you increase this concentration if it keeps on increasing like this, now there will be a point you will observe, you will see that surface becomes water loving to become water hating. So it means eventually the droplets of water will be more like this on this substrate. But then that will only happen when you very carefully monitor the surface angle on poly-D-lysine surface by giving concentration. The reason I am covering this topic in this way, because most of the cell culture which is done currently across the world, barring aside few labs, very few means you can pretty much count them in the fingers, most of them do not take these aspects into account. And that's where comes a conflicting results between different groups, because there may be glass substrate where there may be certain kind of attachment of these molecules. There can be other glass surface which has not been really kind of processed properly. They may have a different kind of attachment, and based on those attachments the contact angle will change. And since the contact angle changes, the cell attachment properties also changes, because you have to realize the medium what you are putting out here is a salt solution. It's filled with salt and other molecules, but the base is aqueous. All the mediums which are being used for cell culture are on a water base, right? If it is on a water base, the problem is this: the properties of the water upon putting on the substrate will be similar to the properties which will be experienced by the medium along with the cells. So for the cells to attach, you have to have reasonable amount of adhering site and a thin film of water around it. So that's why I'm kind of covering this topic in a slightly different way, the things which are not being done. But at the industry level, or at the level where you really have to have a perfect quality control, there you have to ensure that these things are being followed. So what all we talked about? We talked about that you have to do an atomic force microscopy as first technique to know the surface. Second, you have to do a contact angle measurement at the second level of test. The third thing what you have to do is, now when you are putting any compound on the substrate, whether it is getting adsorbed or whether it's forming a thin film or it is forming some kind of chemical coupling, it is always a good idea to do a surface chemical analysis. Okay. What I meant by that is, you have to know that what all atoms are exposed on the surface of that material, or what all elements are on the surface of it. So if you look at poly D lysine, from top you can see you will have carbon, you will have nitrogen, of course you will have hydrogen. These are mostly what will be seeing, and of course you will have oxygen, right? So now doing a surface analysis, and one of the easiest and most thorough surface analysis what you can do is XPS. XPS stands for X-ray, photo, electron, spectroscopy. With few slight changes, we'll continue from here. Okay, thank you.